What's up, AP Bio? It's your boy Aaron, and we have special guests. Miss gets a water cup and fills it with Sprite, Mia Brewer, and we got Krispy Kreme, aka Mr. Jean Jacket, aka been to North Korea and back. Krishna Wreck-It Ralph too. Anyway, we're talking about DNA replication today. So first of all, we're going to start it off. We have the entire list of terms that you will need to know when discussing DNA replication. And then let's actually get to the model. So this is the complete overview of everything you'll be seeing. So starting off, we're going to look at all of the components that make up the process of DNA replication. This is a particular sequence in a genome at which replication is initiated.
DNA is a semi-conservative, meaning that one parent strand is passed on to the daughter helix of the DNA, combining an old and new strand. This replication process begins with helicase, an enzyme that utilizes ATP to unwind the DNA, cutting and separating the DNA strand in half, as shown in the picture. After the helicase unwinds the DNA, single-strand binding proteins attach to the two halves and keep the DNA molecules separated. This happens because the two sides are attracted to each other, and the proteins do not allow that.
In order to help it get started in its process, as RNA primer is built upon the parent strand. Oh my god.
Come on, we can now.
Okay, we'll keep going. At the base of the replication fork, the enzyme responsible for building that RNA primer is RNA primase.
Following the DNA polymerase, the replication fork is formed with a leading and lagging strand. The DNA is anti-parallel, meaning the two strands run parallel but run in opposite directions. One strand runs through 3' to 5', and the other strand runs through 5' to 3', referring to the DNA phosphate groups and sugar backbones. The leading strand is the strand with 5' to 3', and the lagging strand is synthesized in the 3' to 5' direction, or the opposite direction as the movement of the replication fork.
After all this, we are left with two identical DNA strands.